Can everybody hear me? First, I'd like to thank the organizers for inviting me to give a talk here. I've only been very briefly at previous RECOMB meetings, but it's a really exciting group of people that I hope to interact with more in the future than I have in the past. We hear a lot of exciting stories about the discovery of genes and regulatory elements. For instance, we have heard already today several times mention of the ENCODE project, which leads to the identification of large numbers of information encoded in the linear genome. The point I want to make today is that in order to understand how genomes actually work and how genes and regulatory elements work together, we not only need to know all the information that's encoded in the DNA sequence, we also have to understand how that sequence is folded and organized, physically structured inside the living cell. So that's a problem that I call the chromosome folding problem. So if you open a textbook to look at structure of chromosomes, this is one picture that many of us are familiar with, starting with a DNA helix, ending through a number of folding intermediates into something that's recognizable as a mitotic chromosome. I believe that most of these structures drawn here on this picture actually are probably wrong, or at least largely incorrect. And in fact, I think we can agree on only a few of these structures. We have a DNA helix, we have a chromatin fiber, and there's a chromosome here. In between, there's really a large black box of higher order chromosome structures that are poorly understood. The goal of my lab is to try to fill up this black box and see how chromosomes are organized. Chromosomes can be studied using a variety of methods. Classically, it is done by imaging. Here's a picture of a single human genome, or a nucleus. And stained here is one single chromosome. This has led to a tremendous amount of insights into how chromosomes are organized, but not really at the same level as we are all now used to in terms of looking at genomes and finding genes and regulatory elements and so on. Those things happen at a much finer scale than can be seen by imaging. So over the years, we have pioneered a very different approach to studying chromosome structure that's based on chromatin interaction mapping. And the idea behind this approach is that you can view more abstractly a genome as a network of interacting loci, where loci on a single chromosome will interact with other loci on the same chromosome. It will also interact with loci on other chromosomes. So if we can identify for every locus in the genome other loci from the genome that it's physically close to, and we can do this for many, many loci, we can kind of reconstruct a three-dimensional picture of the genome. And as we will see during the talk, this can be helpful in understanding gene regulation. So for this, we needed to develop a series of methods that allow us to take a really close look at these long-range and interchromosomal interactions between loci. As many of you may be familiar with, one technology that we developed a number of years ago is the 5C technology, which is one of the first high-throughput methods to probe long-range interactions in the human genome. The principle of the method is as follows. It's based on the classical 3C method, which starts with cross-linking cells, just like you would do in a chromatin immunoprecipitation experiment, which leads to linking proteins to DNA and proteins to proteins. So what we're basically doing is we're capturing, freezing all the long-range interactions throughout the genome inside a particular cell population. The challenge then becomes to identify pairs of loci that become cross-linked, and this is done through a series of molecular steps that starts with restriction enzyme digestion of the chromatin, and then the most critical step is this ligation step, where two cross-linked DNA fragments are ligated back together to form these unique hybrid molecules that represent long-range interactions. So what we've done here is we converted the very difficult problem of the spatial organization of a genome into a relatively simple problem, which is the composition of a DNA molecule. The 5C method uses a particular detection technology to interrogate the formation of these long-range interaction hybrid products using specifically designed probes that anneal side by side across these unique ligation junctions. And when they do so, we can ligate these probes together and PCR amplify them using the common primers. This is somewhat similar to a PCR reaction. It's called ligation-mediated amplification. The advantage of this method is you can mix thousands of these probes in a single tube, so you can multiplex it at very high levels, and then you can just deep sequence the resulting products. So for instance, what you can do is if you're interested in long-range gene regulation, you can pick 1,000 genes in 1,000 enhancers. And in a single assay, you can map all the long-range interactions between these enhancers and these promoters. I will tell two stories today about how we applied this methodology to look at the three-dimensional structure of chromosomes and the things we have learned from this at two different levels. One is a large-scale domainal level of chromosomes. The second part will be about long-range gene regulatory interactions. So the first part is a story we published earlier this year with Edith Hirt's lab in Paris, particularly her graduate student, Elvash, and a student in my lab, Brian LaJoy, worked on this project. What we did here is we looked at a very specific domain in the mouse genome. This is a 4.5 megabase domain containing the X chromosome inactivation center. The rest of the story will all be about this particular domain, but the results we found apply to all chromosomes. It's just that this is a very interesting biological locus, and Edith Hirt's lab has been studying X chromosome inactivation for many years. So it's a large domain, 5 megabase. There are lots of genes in here. The exist locus, which initiates X chromosome inactivation, is located right here in the middle. We performed a 5C experiment to map the three-dimensional structure of this 5 megabase domain in mouse ES cells. And I'm going to show you a number of these interaction heat maps that are these two-dimensional heat maps that look like this, which is really an interaction map, the frequency with which loci interact, where we plot the locus on both axes. So a particular point in this heat map will be a contact frequency between two loci in this 4.5 megabase domain. Here we have such an interaction map. This is the interaction map, as I said, in male mouse ES cells. There's a single X chromosome. It's not inactive. And what you can see here is that we have this very strong signal along the diagonal, which represents a very frequent point of contact between loci that are relatively close to each other in a linear genome. Of course, we would expect them to be close in three-dimensional space as well. Over here, you would have interaction frequencies between the one end and the other end of the locus. They're very far apart. They don't interact very frequently. We were struck to observe, and we hadn't seen this before in other experiments, we can see this domainal pattern in this heat map where you have these large squares. This is about a megabase domain over here, where all the loci within this domain have a high interaction frequency with each other, but then they don't interact very frequently with the next domain. We call these topologically associating domains that are defined as follows. All the loci within one domain interact with each other quite frequently. They can be hundreds of kilobases in size, and they are separated by sharp boundaries. Okay. Of course, I'll just go pretty quickly through this, but it's a tremendous amount of work that Elvash did. If you get an experiment like this, with an interaction map that's more like a statistical population average, a contact probability map, you need to do experiments to make sure that your interpretation of this interaction map is actually correct. So one interpretation we had is that all the loci within one domain should frequently interact and therefore be close together in space and be separated from the flanking domains. These two domains shouldn't mix. So we spent a lot of time doing imaging, both 3D fish imaging to measure distances between loci, as well as super resolution imaging to make the following points. If you look at loci that are separated by a given genomic distance, but they are located in the same domain, they're closer together in 3D space by imaging, as when you look at two other loci separated by the same genomic distance, but are in two neighboring domains. They are farther apart in 3D space. If we stain individual domains with different back clones, we can see that they're totally separated from each other. They do not mix with each other. Whereas if you take probes that contain a boundary of two domains, you start to see mixing. So the interpretation is, we believe, really correct, that these domains represent little blobs of chromatin that are separated and non-mixing with flanking chromatin. What determines these domains? Well, if you look at histone modification patterns, they also occur in large domains along the chromosome. For instance, if you look at H3K27 trimethylation, often it's not just a punctate signal you get, but there are stretches of hundreds of kb that can be occupied by these modifications. And it turned out that we compared K27 trimethylation and K9 dimethylation with these domains on the X chromosome, we could see that these domains perfectly correlated, not in all cases, but if there is a domain of K27, for instance, it would fill up the entire topological domain. So we thought maybe these histone modifications actually determine the domain. This turns out to be not the case. If you look at mouse cell lines, mouse ES cells that are deleted for enzymes that deposit these histone modifications, by Chip, Edith's Hurt lab had previously shown these modification patterns are completely gone, the domains are intact. That means that the histone modifications follow the domains, but are downstream of it and do not determine chromosomal domains. In fact, it seems that these enzymes just fill up with depositing such a topological domain with a particular modification. So maybe the domains would be defined by genetically defined boundaries. It would be a new type of genome structural element that we hadn't discovered before. Here we were lucky that we had actually picked to choose the exist locus, which is one of these mouse loci that people have studied for a very long time. And therefore, there are lots of resources out there. One particular resource that was useful for us is a deletion mutant of the exist locus itself, which is located right here at this boundary of these two domains. This is a line made by Joost Gripnau's lab, and they were happy to share these cells with us. So here we have deleted that boundary, and we repeated the 5C analysis of the domain. And what you start seeing is that the two domains used to be completely separate from each other. But when you delete this boundary, they start mixing, but new boundaries seem to emerge. So two things we conclude from this. One, we think there are genetically defined boundary elements that prevent mixing of these domains. We do not know how these elements would do that. And second, there's clearly something we don't understand here because the domains don't completely mix and new boundaries seem to emerge. What happens to these domains when these ES cells start differentiating into different cell types? So here we have the contact map of undifferentiated ES cells. When these cells are differentiated into neural progenitor cells, we see that domain structure remains intact. Even though gene expression, as I'll show you in a second, changes dramatically. Including, if you look at embryonic fibroblasts from mouse embryos, you can still recognize the domainal organization of these cells, although the internal structure of these topological associating domains seem to change. So this domain is still recognizable in these embryonic fibroblasts, but there's clearly something happening internally. So we like to think that these boundaries are constitutive, but the internal organization changes. And as it will come in the second part of the talk, I believe this is related to the occurrence of specific long-range interactions between genes and regulatory elements within these topological associating domains. We looked at the relationship between these structures and gene expression. So a student in Edith's lab followed by real-time PCR the expression of all the genes in this domain through 18 time points during ES cell differentiation. And she showed that genes located in the same topological domain are highly correlated in the way they modify and change their gene expression over time during this differentiation pathway. This is not to say that these genes all have the same expression level or always on and off at the same time, but the trends of going up and down during this timeline is the same for these genes within domains. Okay. While doing these experiments in parallel and independently, Bing Ren's lab had done a lower resolution Hi-C study of the mouse genome as well as the human genome and identified similar structures as we had found on the exist locus. And in fact, his data correspond extremely well with our data for the XIC locus. And we conclude from these two stories, they published their paper back-to-back with us, we conclude that this kind of domainal structure is really uniform throughout the mouse and human genome and possibly other organisms. I think there's data in flies as well as in some unpublished other data sets. I want to just remind you that the fact that in the genomes there are blocks of co-regulated or co-expressed genes is not new. About a decade ago, several groups reported by analyzing gene expression data that groups of neighboring genes show very correlated gene expression across tissue types, for instance. I believe, we are currently looking into this in more detail, that this could be related to those topological domains that we discover here, which would give a structural explanation for this phenomenon that has been seen many, many years ago. So to go back to the original question of how chromosomes fold, I'd like to think that we've added at least one level in this hierarchy that we call TADS. It's a pretty large-scale structure. It's about, on average, it's about 800 kilobase. But this domainal organization into structural domains that also have functional consequences for gene regulation. So let's look at a little higher resolution, and not looking at large domains, but look at individual genes and regulatory elements. So we're very much interested in this aspect of gene regulation and how it is impacted by three-dimensional structure of chromosomes. If you look at current ENCODE data, for instance, this is just a drawing of it, but there are, whatever, 25,000 genes and possibly 1 million or so regulatory elements. So every gene finds itself surrounded by a tremendous large number of regulatory elements. And the question is, how can we predict or identify which regulatory element actually affects which gene? This is not a trivial task because regulatory elements, as we have heard this morning from Len Panaccio's talk, can be located sometimes at very large distances from their target gene. So the nearest gene for this element, for instance, the nearest target gene may not be the nearest gene. It could be this gene over here skipping over these genes. So currently, it's very hard to predict these relationships, and instead we probably just have to measure it. So what we set out to do is to look at a large set of genes and to measure these type of three-dimensional structures where regulatory elements would directly physically associate with the target gene. We've known from many cursory studies and single gene studies that these kind of structures are formed to regulate genes. So if we were hoping to do this for a large number of genes to do two things. One, we can identify for those genes which regulatory elements they interact with and possibly are regulated by. But hopefully we'll learn something more general about the rules that underlie these long-range relationships that may at some point help us predict these relationships. So this was actually part of the ENCODE project, and this was the ENCODE project in my lab. Really 3 extremely talented people were involved in this. Amartya is a postdoctoral fellow. Brian was also involved in the first part of the talk. He's leading all the bioinformatics in my lab. And Gaurav is also a computational scientist. So we decided to look at not the whole human genome, but to look again using the 5C method to look at very high resolution structures in the ENCODE pilot project regions, which were picked a number of years ago. These are 44 different regions that are scattered all throughout the genome, in total covering about 30 megabase. Now we changed the 5C mapping a little bit to specifically look at long-range gene regulatory interactions. So in this case, we chose 628 transcriptional start sites located in that 30 megabase of the human genome, and we designed primers for the start sites, and we designed the second set of primers in such a way that we would interrogate interactions between these start sites and all the flanking chromatin that are not a start site. So what we do in this experiment, in a single experiment, we will get an interaction profile for each gene with its flanking chromatin. Okay? And then we hope to find, for instance, a locus over here that would specifically interact with this transcription start site. Okay. We did this for 4 of the ENCODE cell lines, K562, GM12878, this is a lymphoblastoid line, HeLa, and human ES cells. As I said, we interrogated 628 genes versus a little over 4,500 distal sites tiling all through the 30 megabase. This seems like a relatively small experiment, but just by nature of these type of interaction studies, the number of interactions you're interrogating per experiment is quite large. Here's an example of one such a long-range interaction map. It's in fact only for a subset of the ENCODE regions. Every row here in the heat map is a single transcription start site interacting, in this case, with about 15 megabase of genomic DNA. Now, these squares you see here are not topologically associating domains. These are actually individual ENCODE regions. The reason why they show up like this is that most start sites interact mainly with the chromatin surrounding that start site in the same region, but don't interact with the other ENCODE regions, which often are located on different chromosomes. Here we have an instance of some interaction between this ENCODE region and that ENCODE region. They happen to be on the same chromosome, chromosome 7. Here we have one of my favorite loci, the beta-globin locus. It's a 1 megabase domain. There are lots of start sites in there, not just for beta-globin, but also for several other genes. And here's indicated the one interaction profile that I'm going to show you a little in more detail, that of the gamma-delta genes in the beta-globin locus, which are highly expressed in K562 cells. And I'm going to show this as an illustration of how we're analyzing this type of data to identify significant long-range interactions. So we can take this individual profile for this one gene and plot it as a regular graph, and we call this our gold standard because in this we know a lot about this interaction profile, and we wanted to make sure that we are actually capturing those long-range interactions that are very well described in literature. So it's a 1 megabase domain. We're plotting it here. There are lots of genes here. I didn't name them all, but the gamma-globin genes are right here in the middle. And here's just the raw interaction profile in K562 cells for this gene. So in general, you see very frequent interactions nearby, and it decays with genomic distance, very similar to this diagonal that I showed you before in these heat maps. So, and there's signal everywhere. There are actually no zeros in this map. What do we consider a long-range interaction? Well, we took the entire data set obtained for this particular cell type as a training set to learn what the general background function would be for a given transcriptional start site, which is shown in a red line over here with plus or minus 1 standard deviation. So now we have basically a model that says for a given distance we have a certain expectation, and can we now look for interactions that happen more frequently than that expectation at a particular FDR. We picked up 1% FDR, and in green here we indicate interactions between this gene, the gamma-globin gene, and distal sites that are reproducibly found in 2 biological replicates. And it turns out these are all the known regulatory elements for this gene. So these are, for instance, here we have the locus control region, very famous, regulating the gene. It's about 50 kb away from the target gene. There are lots of flanking CTCF sites that in the past had also been found to interact with these genes. All these interactions are non-significant in cells that do not express these genes. Okay, so that all looks good, and we have a way to find significant contacts. We find, in fact, not just these, we find large numbers of chromatin looping interactions in these 4 cell types, thousands of them. By just looking at 1% of the genome, we already found thousands of these long-range interactions. A little over 50% are only seen in 1 cell type, so many of these interactions are highly tissue-specific. We used a lot of the ENCODE data. This is really an example, I think. Our study really tremendously benefited from all the data generated by ENCODE. And we can see that we can now look at the distal sites that we find interactive genes to see whether they have certain features that we associate with regulatory elements. And in fact, in the differentiated cell lines, the lymphoblasts and so on, we find all the features associated with active regulatory elements, K9 acetylation, K27 acetylation, K1 monomethylation, and so on, depletion of K27 trimethylation. But in ES cells, we actually did not see such an enrichment. And in fact, we see some enrichment for K27 trimethylation. So ES cells are a little bit different. We used segmentation analyses really pioneered by the Manolis Keller's lab and Bill Noble's lab. ENCODE generated, segmented the genome in 7 different states. One of them is, for instance, shown here, an enhancer state characterized by a combination of features, such as histone modifications and so on. So now we can look to see whether the distal sites we find interacting with genes would fall in any of these particular classes. And it turns out that we find promoters interacting with 2 different types of enhancers, but also with other things that look like promoters, even though we selected the distal sites to interrogate as being non-promoters, but some of the things have chromatin features that look like promoters. Very strong enrichment for CTCF. I'll come back to that in a second. So now we can classify long-range interactions as those between a promoter and an enhancer-like element, or a promoter and another promoter-like element, or a promoter and a CTCF site. And it turns out if you do that, for instance, for these lymphoblasts, the large majority of long-range interactions are actually with CTCF, but we have enhancers and promoters. There is some overlap between these categories because the resolution of our analysis here, several kilobase, which means a particular restriction fragment of a few kilobase can contain both a CTCF site and an enhancer-like element. So now we can ask more specific questions. If a gene interacts with an enhancer, is that in some way correlated with the gene expression state of that gene? Now it turns out that is the case. If you look at promoters that are having a long-range interaction with an enhancer-like element, the majority of them are expressed compared to those that do not have such an interaction. And in addition, those genes that are expressed and have an interaction with an enhancer are significantly more highly expressed than genes that do not have such an interaction. So these are two things. One, it says that these interactions may be related to regulating that gene. It also, of course, is a nice validation of both the enhancer state and the 5C analysis. None of the other categories of long-range interactions seem to correlate with gene expression. Again, ES cells were an exception. In ES cells, we do not see such a correlation between interacting with a distal enhancer and gene expression. Now, I must say, if you look closely at the segmentation state of enhancers in ES cells, they actually are different than those of differentiated cells, because these enhancer-like elements in ES cells, according to this segmentation, look more like a poised or inactive enhancer. So the final analysis we wanted to do is, now that we have found these long-range interactions, can we look to see whether there are some generalities in terms of, if you look at a gene, whether it interacts with upstream or downstream elements, how far away from the gene can we find long-range interactions, and so on. So what we did is what we all do very regularly. We plot up all the transcription site sites here in the middle, pointing to the right, and plot the distribution of these long-range interactions around the promoter. And these are just, in yellow we have enhancers, and in blue here we have CTCF sites, and in red with promoters. What was striking is that, and this is for all classes of long-range interactions, that there is this general decay with probability of having a long-range interaction with genomic distance. This is minus 750 and plus 750 kilobase. But there is this asymmetry that there's a tendency to have interactions with elements about 100 kilobase upstream of the gene. Oops. And this is particularly clear if you split this out in those transcription sites that are expressed versus those that are non-expressed. I must say this non-expressed is really a lowly expressed data set. So there's this interesting asymmetry. And it turns out that most of the long-range interactions are actually happening just within a few hundred kb upstream of the gene. This might at some point become helpful if we want to predict target genes for elements. One final analysis that comes up a lot when I talk about these things, and that is, what's the role of the CTCF protein, which has been classically described as an element that would block enhancer-promoter interactions. So can we actually see whether a site bound by CTCF in a given cell type would somehow prevent a long-range interaction between an enhancer on one hand and a promoter on the other hand? It turns out that is not the case. Over 80% of our long-range interactions skip over at least one site bound by CTCF in that cell type. So that tells us two things. Well, it's of course possible that CTCF occupancy is far lower than we believe, but it means that, in my mind, that the majority of these CTCF sites actually are not the classical insulator protein. That's not to say that all CTCF sites, that this would apply to all CTCF sites. There is a set of CTCF sites enriched at the boundaries of these topological associating domains. They clearly insulate chromatin in two domains, so that a subset of CTCF may actually act like this. And this just brings me to the final point to bring the two stories together. What's the relationship between these topological domains and these long-range interactions that we discovered? It turns out you can expect probably that interactions between genes and elements would probably happen most frequently within these domains, very rarely between the domains, and that's exactly what we see. The large majority of our long-range interactions happen within these domains. Okay. So what I showed you is that we see abundant looping interactions between TSSs and distal elements. They look like CTCF site and insulators and enhancers, but I must say there's a large class of unclassified elements that I would invite all of you to look at, and hopefully maybe there's an unanticipated class of elements we have missed. We see this interesting asymmetry in the looping landscape with a bias for things upstream. CTCF sites do not block this looping. And one final point that didn't make it to the paper, but I think is very important, that ES cells seem to display an additional class of looping interaction between genes, and we think enhancers that look like poison enhancers. So I hope we may have filled in another layer in this hierarchy of chromosome folding. There must be more layers, and there must be a lot of details we're missing, and some things are probably wrong. But this is the direction we're going to, trying to really understand folding of chromatin from a very local scale to a very large scale. These are the people involved, and I'm more than happy to take any questions.
Time for a couple of questions, and I'll request the next speaker to please come up and set up.
Yeah, thank you very much for the exciting talk. Should we think of every enhancer as being involved in looping interaction? In other words, are there some that might simply be acting locally, or should we think of every interaction that we're not currently finding as missing in these maps?
It's a great question. Sometimes I'm delusional, and I think nothing in the genome makes sense except in 3D. But for sure, that might be wrong. You're right. There may be elements that actually do not act in this way at all. There are plenty of enhancers in the ENCODE regions that we interrogated, but we never find looping for.
Do you think that could be a condition?
The enhancers are active in that cell. We thought about it. It's possible that the target gene is outside the region that we interrogated. That's possible. But actually, it's a good idea. We have never really went back and said, is there something different about those?
And just to get a ballpark, what percentage did not find any evidence for?
At least half.
Okay, thank you very much.
Do we have data on, say, human mouse conservation of looping at the exist locus?
I don't think so. It would be interesting to have that. We have mouse-human comparisons, and Bing Ren's group also did it, to look at the domainal level. At that level, the conservation is very, very high. But for looping, for exist, I don't know.
No problem. And last question. Obviously, ENCODE regions, the 1% were selected to reflect different gene densities. So do we see generally different looping landscapes in, gene-rich, gene-poor regions out of the 1%?
It's a good question. The problem is, we have a problem here with the way we designed the experiment. So if a region is very gene poor, we just have very little data for it because we anchor them on start sites. But it's a great question. We do know that chromatin in gene poor regions looks structurally different from that in active regions. But we have not spent a lot of time on it. But it's a very good question.
We'll do one more question. And Job, could you let the next speaker set up while you're answering that question?
I was wondering whether you look at motifs or other specific sequence elements that lay at the boundary of the domains you identified.
Say that again?
I was wondering whether you have looked at motifs or other sequence elements that lay at the boundaries of the domains you have identified.
Yes. So the boundaries actually, and this could be a limitation of the methodology at this point, the boundaries are typically 10, 15 kilobase. So these are this is a bit fuzzy. It's not that we can find one element. Bing Ren's group and we also have looked at to see whether certain things are enriched near the boundaries. CTCF sites are enriched. Any transcription site actually is enriched. Almost everything you look for is enriched near those boundaries. So I think we have a big problem in our hands here because as soon as you normalize for gene density, the enrichment for CTCF goes away. So these are very highly correlated data sets, so it's difficult to tease out which is the cause of the growth.
Let's thank our speaker once again.